should thank our organizers and our hosts for this wonderful conference. I'm going to talk about genetics today, and I'm also like a motherless child here. I think I'm the only geneticist. My talk, I'm afraid, will have terms and concepts that may be unfamiliar to people, and I'll do my best to explain things as I go. But if I way out of line, please raise your hand. Ask me what I'm talking about. I'm capable of losing hundreds of people at the same time. I'm going to talk in this talk about the so-called dopamine, E4 dopamine receptor. I'm going to talk about it because I think it is something that will be important in the future. Human ethology, sociobiology has steadfastly ignored differences among people, or even the possibility of gene differences among groups of people. But with new evidence, new data coming from molecular biology, I think we can't afford that luxury anymore. So I'm going to suggest some things about this. Nothing I'm saying here has been well demonstrated. But my sense is that this and other systems over the next 5 or 10 years are things that all of us are going to have to try to understand and cope with. So here's some of the jargon, some of the terms I'm going to use. I'm going to start out with this. I'm going to talk about ADHD. It's attention deficit hyperactivity disorder. It used to be called childhood hyperactivity. This is epidemic in North America. It's not clear whether behavior has changed or whether our public education system has changed, but many children suffer from this problem. CAD is jargon. A CAD male is a male who abandons females. Dopamine is a neurotransmitter. It's a substance in the brain that's involved in communication between neurons. It works because receptor molecules pick it up and move it into the cell. There are 5 dopamine receptors, and I'm going to talk about genetic diversity in the 4th one. So it's called the D4 dopamine receptor, or DRD4. VNTR stands for variable number of tandem repeats. If we look at sections of DNA, in our cells there are parts of the genetic material, usually in junk DNA, non-coding DNA, where there are stutters. There are sections of code that are repeated many times. Short repeats are called SDRs, or microsatellites. In general, VNTR. A VNTR locus stands for variable number of tandem repeats. So it's a common, easily typed kind of genetic diversity. I'll mention coalescent. If we have a sample of genes from a population, if we could follow their history backwards in time, all of these genes are from the same locus and different organisms. This history will be a tree. They will all derive ultimately from a common ancestor in the past, and that history is called a coalescent. And finally, don't try at all to write down anything here. As soon as I find an internet connection, I'll put both my talks on that website. It's just a computer at my house, and anyone can access my talks over the internet, so you don't need to try to copy things. All right. Well, D4 is one of several receptor molecules for the neurotransmitter dopamine. There is a repeat polymorphism in the gene. In the coding part of the gene, there's a VNTR. And in this part of the gene, you may have 2, 3, 4, up to 11 copies of a 48 base DNA motif, it's called. And these code for the protein product. So each 48 base DNA motif codes for 16 amino acids, and these are actually put into the functioning protein. Now, if you know some of the history of psychiatry and psychobiology, you know that all of these neurotransmitters, as soon as anyone found a way to assay them, all over the world, psychiatric patients were bled, exsanguinated, paper after paper trying to correlate levels of various brain chemicals with psychiatric disorders, and looking at all of it, years and years of it, gallons and gallons of patients, giving up gallons of blood. There's not much to it. There's nothing to it. The correlations between quantity and behavior aren't much. But it turns out that this gene, that genetic differences among individuals are pretty reliably correlated with behavior differences. People who have the 7R, in other words, the gene with 7 copies of this insertion, are much more likely to show novelty seeking. Now, when they talk about showing novelty seeking, it means they gave them some pencil and paper test, and I don't know what they're worth, and I don't believe them. But they also are at increased risk for being hyperactive children, being diagnosed with a strict form of attention deficit hyperactivity disorder. Now, 7R is the only common long gene. The other ones, 9, 11, are quite rare, and they may also be associated with behavior change. It's not at all clear, there aren't enough data. But 7R is quite common in many populations, and the association with childhood hyperactivity has been replicated many times. So, attention deficit hyperactivity disorder, ADHD, is inappropriate inattention, impulsivity, and high levels of activity, high levels of energy, with onset before 7 years of age. Now, if you notice this, part of the definition is inappropriate. And what's inappropriate? Well, it means, in the context of most of the literature, it's inappropriate for elementary school classrooms in North America. But these are high energy children. They're very active. They don't pay attention. They're impulsive. I don't believe for a moment that impulsive is the right word. I think they're unpredictable. I think they're very difficult to deal with personally. It's very difficult to predict what they'll do. They, among other things, they lack empathy. We've had some discussion here yesterday and the day before about empathy, and a conspicuous characteristic of these children is that they really seem to lack a lot of empathy. there's a clear relationship between childhood hyperactivity and adult sociopathy. Adult sociopaths are essentially people lacking what we call conscience. And they have many of these characteristics of hyperactive children, impulsivity and unpredictability. And 7R alleles increase the risk, about doubles your risk of being diagnosed with strict ADHD as a child. Now, that will let us read the words quickly. And if you're not a native English speaker, you may not get the point, but what happens is if you sit school children down and give them a task, and the task is to read the word as quickly as possible, and if you show them the word blue in the green color, it confuses them, and they're delayed. If you show them the word yellow with red letters, they're delayed. This is one of the specific neurological deficits often associated with childhood hyperactivity. A lot of hyperactive children seem to have neurological damage of some sort. They seem to have an IQ loss and troubles with some of these tests. But children with 7R alleles in the DRD4 locus who are hyperactive have no trouble with these tests. They have no neurological deficits, and they seem to enjoy an IQ gain. All right, so there's also evidence now that childhood hyperactivity, there's at least, there are probably many different kinds, but some of them seem to have suffered what most of us would call a damage or disorder. But children who have these 7R alleles, 7R genes, and have childhood hyperactivity, they're doing fine. They don't have any trouble reading the word blue and green letters, and they seem to have an increase in anything in IQ. Am I going too fast for the students? Does it make sense?
Yes.
What I'm saying? Okay. Well, this is, for an audience of geneticists, all of you would look at this very carefully, and you don't need to pay any attention to it at all. It's a diagram of the gene. Exons are parts of the DNA that are actually turned into protein, and this shows that the 48 base pair VNTR is in exon 3, and down below there's a diagram showing world frequencies. The common ones, as you see, are 2R, 4R, and 7R. What those motifs are shown here. None of us can read it. It doesn't matter. If you're dying of curiosity, download it from my website, blow it up, and you can stare at it to your heart's content. It's nonsense. But here's something what's known of evolutionary relationships among these genes. The ancestral state seems to be 4R, and the motifs are numbered 1, 2, 3, and 4. And it's very easy to take one of these genes, have an unequal crossing over, and end up with a 2R. It's very easy to have unequal crossing over and a mutation, get a 3R, a 5R, a 6R. These are all easily derived from 4R by mutation and crossing over events. The minimum number of steps needed to make 7R is, I think, 7 steps. In other words, very difficult, highly unlikely, highly improbable event. Very difficult for nature to start it out with a 4R and make a 7R. It's multiple steps. So if we look at relations among genes at this locus in the population, there's basically two kinds. There's 4R and its relatives, and there are the 7Rs. Now, these are hypothetical diagrams of gene history. In other words, we took a sample of 5 genes from 5 people in the population and traced their history into the past. And they have a tree-like history. This is called a coalescent tree. The depth of the tree, in theory, is in generations 4 times the size of the population. There's a lot of population genetic theory about this. Most nuclear coalescent trees from humans are on the order of 1 million years old. But in some cases, trees look like that tree on the right, with a very deep top root. Too deep for it to have just evolved neutrally. One example is ABO blood groups. Chimpanzees have ABO blood groups like we do. So we know that the top of the ABO coalescent tree has got to be older than 7 million years. It may be much older. So if we could diagram a history of genes from the ABO locus in this room, we'd have perhaps A's here and O's here, and the common ancestor, perhaps 15 million years ago. The difference is maintained by selection. It's called a balanced polymorphism. Nature is keeping both kinds of genes present, and one diagnostic of this is very deep coalescent trees. Another familiar example is the HLA system, the immune response system, the system that's typed for tissue transplants. The top of those gene trees is on the order of 40 million years. So I may have 2 HLA genes, and one of them may be more closely related to one in an orangutan than to the one on my other chromosome. So the history of HLA is very deep. Again, it's maintained by balancing selection. Once a type becomes rare, it enjoys a selective advantage. So this is a signature, a way that we can look at a sample of DNA and ask ourselves whether this looks like the evolution of junk, neutral DNA, passive DNA, or whether it looks like there's selection going on. Here's another signature of selection. This didn't come out very well, but what we did here is we took a sample of, 12 or 15 genes. And this is the history. Right here, this one suddenly became very successful. It was positively selected. For some reason, this particular form at this time suddenly had a selective advantage. It spread very rapidly at the expense of the others. So what we have here is I can just reach. What we have here is the most common type of gene. They're all the same. Those hypothetical round dots are mutations. The probability of a mutation depends on time, on the length of the tree. And this thing has spread so recently, there's been no time for mutations to occur. Every single copy of this gene is the same, while, for example, this one and this one differ by 2 mutations, right? Because if we follow them back to a common ancestor, there's one mutation, there's another. But these are all the same, so something is common, but there's no diversity within the class. It means it's spread very recently. And this is what 7R looks like. This 7R gene is very successful. There's no diversity in it. Everyone in the world is the same, exactly the same. They have a very recent history. They've been spreading like wildfire. But it's been around for millions of years. It's been around at very low frequency, and it's become successful very recently. So again, let me just say it again. This is supposed to represent 7R. If we compare two 4Rs, if we pick two genes from this room at random, they're probably 4Rs, but they'll be different. There'll be mutations between them. 7Rs are all the same. Here's some data on the world distribution of this 7R. These are gene frequencies. So if we look at Kalahari-Bushman, we collect a sample of Bushman DNA. They have no 7R, 0. Now remember, Bushmen are the subject of one of the most famous ethnographic monographs of the century, a book called The Harmless People. They're gentle, laid back, mild people. They have no 7R. Pygmies have 15%. Bantu-speaking people in Africa, 19%. On Europe, in average of studies from continental Europe, about 16% of the genes at this locus are 7R. There's none on Sardinia. And I mention that because we know that most of the population of Europe is descended from a wave of advance of Neolithic farmers starting after the end of the Pleistocene, starting 8,000 or 10,000 years ago. As farming spread across Europe, it brought new genes with it. The populations that seem to be the closest approximation we have to pre-Neolithic relics in Europe are the Basques, the Pyrenees, and Sardinians. So it may be significant that there's no 7R in Sardinia. European Jews, just like the continent, 16%. Sample of Jews from Yemen, none, 0. Nazioi, Melanesia, 30% of the genes are 7R. New Guinea, 25%, 17% of Malays. Samoa and Micronesia, very little, and especially in Samoa, none. In Asia, groups on the edge of Asia had a little bit, but in large samples from China and Japan, none. 0, 7R. In North American groups, 26%. Central America, 39%. The South American average is 63%. There's some groups involved in South America where the gene frequency is 75%. So the other famous ethnography of this century is about, well, in South America, it's called the fierce people. And it's about people with 75% 7R. Well, we have evidence of selection in this very deep group and the lack of diversity within the class. And it turns out that population differences around the world are much greater than they are for most genes. So this pattern here is not an accident. It doesn't look like, if we can take a big sample of genes and look at world differences, they'll be different. They won't be this different. People are too different here. So does it make sense? Pierre, did you have
You're talking of Aboriginal populations in
Yes.
America? Yeah, okay.
Yeah, Aboriginal populations. Well, I don't know if it makes sense. What I'm going to give you now is a hypothesis about it. Why it looks like it makes sense to me. It's untested, but at least we are writing some proposals to test some of this. So it's not all a fairy tale, but it's kind of like fairy tale, but it may be right. But in the meantime, I want to bring up an interesting way of thinking about human differences and carrying on some of the themes of Glenn's findings. Like all males, human males sometimes can put their effort, their energy, their risk into mating effort or into parental effort. A mammal, or human, male can put his time and energy into provisioning a mate, provisioning an offspring, or he can put his time, energy, and risk into competing with other males for access to females. And if we look for this pattern in societies, I think we can see it in the jargon, the shorthand that I'll use is to talk about dad and cad societies. So a society where males put their effort into mating, we'll call cad society, and we'll call dad societies where males are parental. And if we look around the world at this, there are certain regularities, and I want to talk a little bit about these. Some regular patterns. Societies where males are parental, in other words, where a guy gets up in the morning and he does something to feed his children, tend to be foragers, hunter-gatherers on the one hand, and dense agriculturalists on the other. Labor-intensive farmers. And in complex societies, they're what sociologists call working class. In between, in societies where, especially where there's a lot of shifting cultivation, slash and burn agriculture, the economists call these female farming systems. And when economists call female farming systems, this is a euphemism, this is a polite way of saying these are societies where men live off women, where women produce food and men parasitize women, basically. And all over the world, in low energy agricultural systems, females do the production, and men, men are tough guys. They put on paint and they put on weapons and they take drugs, dance, and go raid the next village. So associated with mating effort is local raiding and warfare all over Highland New Guinea, all over lowland South America. What we males do is sit around and discuss our next raid and wait for the women to bring us food. In societies where males are parental, there's plenty of war, but it's often external warfare. We recruit the males, put them in the army, and send them to fight somewhere else. Or there's enforced peace. Mating effort, cad societies are associated with matrifocal households, mother-child sleeping arrangements. In many of these societies, the men live in a men's house, and they don't live with families. In parental effort societies, a good sign of this is just look where people sleep, where men and women sleep together. They're often a parental effort society. Mating effort societies are characterized by sort of male-female hostility. In these societies, typically women just wish that men would go away and leave them alone. But what men do is protect them from other men. But certainly the idea of any kind of affectionate relationship between males and females is regarded as absurd. In many of these societies, there's a well-developed, what's called fear of women complex. The idea that women are dangerous, they'll pollute you, they'll take your vital bodily fluids, sap your strength, and that men have to stick together and protect themselves from the women because the women are going to take over society if they don't. Well, in the old, in the Bible, in the Old Testament, we know what Delilah did to Samson, right? That theme, women take your strength, is most developed in these societies, in parental effort societies. Relations between the sexes are much more likely to be intimate and egalitarian. In these mating effort societies, these are the ones that are most likely to have harsh male initiation rites. As Glenn was talking about this morning, males are raised by women, they're rather feminized, and then the men get a hold of them and turn them into sort of fierce warriors with all their paint on them. Well, in mating effort societies, there's lots of violence, crime, male belligerence, machismo, protest masculinity. Males are likely to be tough guys, and they're likely to have paint and decoration, beads and lipstick and makeup and fancy weapons. If you look at a Yanomama warrior, if you look at Plains Indians in North America, they were looking good. They had lots of feathers and decorations and all. If you look at a Chinese peasant or a Kalahari bushman, they don't have any decorations. They're just out working for a living, producing food. Male oratory and display are characteristic of these male competitive societies. The North American Plains Indian chief gets up and he gives a 3-hour speech, that sort of thing. Male competitive societies tend to be polygynous, transient matings, pair bonding and monogamy are much more characteristic of societies where males are parental. In these societies, often children are left to the peer group to take care of them. There's not so much parental involvement in child rearing in these societies there is. The extremes of parental societies seem to be societies where there's dense agriculture, peasantry, and in these societies the scarce good for reproduction is often males with their land. When males and male land are scarce, females have to compete for males. The way females compete seems to be on the basis of virtue, purity, and chastity. So in societies where males are the scarce resource, in societies where we find things like females crippling themselves, female circumcision, veils, all kinds of control of women. In male competitive societies, we don't see any of that. We don't ever see female circumcision or any kind of display by women of virtue. We see display by males of how tough they are and what good warriors they are. Well, so this is just a slide from a famous film about, this is Highland, New Guinea. These are men from two different groups throwing spears at each other. This kind of thing was suppressed by the colonial government. It's come back in a big way. You can drive around Highland, New Guinea, drive around the corner, and drive right into one of these spear fights today. A colleague of mine a few years ago at P&G Medical School had his two best lab techs go home for Christmas, and they were both killed in a spear fight like this. This is what guys do. And these societies, the women, during this fight, pay no attention. They're out working in the fields. They think this is all silly guy stuff, and they wish that the males quit acting so silly. Here, on the other hand, there are a couple of Kalahari Bushmen. They're out hunting. They have no decoration, no beads, no paint, nothing. I've shown these people pictures, for example, of North American Plains Indians, and they think it's just hilarious that anyone would gaudy themselves up like that. Well, this is an aside for the paleoanthropologists here. Coon Bushmen have been foraging, hunting and gathering in Kalahari. Their technology is unchanged for something like the last 20,000 years. They bring down even giraffe. I've had lots of giraffe steak brought down by poison arrows. They're real good at what they do. And this is the sum total of their technology. Arrows here with poison tips. They look like something a child made. If we go several thousand kilometers north of them, into the Congo Basin, and look at technology, here it is. These are drawings by Henry Stanley. Technology in the Congo Basin. Beautiful, spectacular stuff. This is made by men who are sitting around eating the production of gardens. They're living off women. They're not doing anything, nothing on. And they make these beautiful weapons that are completely useless. They're not anything you ever use. Anyone who's ever butchered an animal would never use anything that's up there. This is stuff you hang on the wall. It is male display. It isn't technology. And I'm putting this up here just because some of us are interested in the spread of modern humans, the Upper Paleolithic. Many archaeologists are very impressed with the elaborate new technology of the Upper Paleolithic. And my suspicion is that it may just be the difference between this and that, one of which is the toolkit of working people, and the other is sort of intensive male nonsense in display. Well, anyway, back to the story. Now, let's go the other way. All right. So here's a history of humanity, 3 paragraphs. There's abusive detail in all of this, right? But basically, many foragers had parental dad males. The exceptions are places where the rich, reliable, concentrated resources, like, for example, the Northwest Coast, where we males may produce a lot of food, but we had to do it a couple of weeks out of the year during the salmon run, say, and the rest of the time we could be tough guys, make totem poles and masts, and have a good time. Low-intensity food production leads to systems of female farming, local male coalitional violence, which leads to a kind of stable anarchy. Lowland South America, Highland New Guinea, population never gets very high because they're killing each other all the time. The violence, the local raiding, the local warfare keeps population down, and it's a stable anarchic system. And what breaks it is polities, is the state, the political organization. Because once you get that, it stops the local rating. Once you stop the local rating, population grows. Once population grows, you have to intensify your agriculture, put more labor into it. Pretty soon, these poor males are back working in the field, with a plow, and pretty soon, Highland New Guineans, they've had to put away their feathers, their paint, and everything, and they're out on the rice paddies, morning and night, working. So, my view of civilization is that it's, the failure of primitive anarchy. Well, finally, if we map what we know about DRD4 onto world social systems, the canonical examples of dad or parental societies are Bushmen, who have no 7R, and they're Asian, South Asian, and East Asian, dense peasant agricultural systems. Where men are drab, dull, they don't give speeches, and they're working all the time. And these are both groups with no 7R at all. There's a probability that there used to be 7R in China and Japan because there is a kind of 2R there that is derived from 7R. So the suggestion is that there's been 7R in the past, it's gone. Canonical examples of male competitive or CAD societies are Amazonia, like the Yanomami, Highland, New Guinea, and in the New World in South America, the frequencies of 7R are up to 75%. Why the New World? So why is there so much in the New World? I suspect it had to do with the absence of diseases and consequent higher levels of a lot of violence there. And Keeley's book on War Before Civilization shows that the New World was an especially violent place to be. Why would one suspect this kind of correlation? Well, what makes you a good warrior? Well, everything that doesn't make you a good parental male. If I'm going to be a good warrior, the last thing I want is empathy. I want to be unpredictable. I don't want to bond with my family. And so if you look at a textbook list of characteristics of people who are called sociopaths, all of them would make excellent fighters, excellent participants in warfare. So I suspect that what this is a nice case of gene-culture coevolution. That cultural differences are selecting for gene variants that affect behavior and affect personality. Once that selection has occurred, it may make it a little bit more difficult to change back. I don't know. But the new technologies in molecular biology that I started talking about at the beginning of this talk regarding how to read from patterns of gene diversity, something about the gene history, I think they're going to open up lots of cases like this, and a few others I'll just put up here that I think it'd be worthwhile to keep your eyes open for. I think these are going to be interesting new cases. One is human luteinizing hormone. Human luteinizing hormone is involved in the regulation of the reproductive cycle of females, in the regulation of testicular function in males. It turns out there are two kinds of human luteinizing hormone. Again, a very deep root They're physiologically quite different from each other. One of them has a much more powerful effect in a short half-life. Males with the second form of luteinizing hormone have delayed puberty and slower growth. These have easily detectable, visible effects on people's phenotype. It's only recently become clear that there are two major kinds of luteinizing hormone genes, very different from each other. Again, this is a system that we're trying to start to look into. Monoamine oxidase, this is a neurotransmitter degrader. Again, it's one of these chemicals where thousands of psychiatric patients have been bled looking at levels of monoamine oxidase. Nothing has ever been found that could be replicated. But all of a sudden, we've been looking at the molecular biology of it. There's two major types. There's one type that's quite common in Europe and Asia that's absolutely absent in Africa. Again, a very unusual pattern for a human gene. I think this will be worth watching in the future. And finally, I'll just mention IGF-2. IGF is insulin-like growth factor. And IGF-2 is insulin-like growth factor number 2. It's been of interest because it's a gene that if I transmit the gene, it turns on in the fetus. It's imprinted. If it comes from the father, it's imprinted in the fetus. If it comes from the mother, it's not turned on. This is called gene imprint. And what it does is it makes my baby grow bigger. So if I'm some mammal and the female has had multiple insemination so that there may be a utero competition between half sibs, then anything that I can do to get my baby to steal nutrients from your baby is going to be favored. And IGF-2 is in rodents imprinted from males. There's a mirror image system in females, the IGF-2 receptor, and all it does is defeat the gene from the male. It soaks up IGF-2 and neutralizes. My IGF-2 receptor is not turned on, the one from the mother is. Very interesting story in rats. It's not imprinted in humans, but people have looked a little bit more, and it turns out that in some humans it is imprinted. There are several major variants of it. It has an effect on birth weight. It's a selfish gene. It sucks more nutrients from the mother than she wants to give it. It has an effect on birth weight, 4 IQ points, and it's a tumor suppressor. So these are 3 systems that have appeared in the literature in the last year or two that I think we should watch. Because I think that our field of interest in the evolution of human behavior, I think we can't ignore human genetic diversity anymore. I think we are going to have to try to understand gene differences, the way we're different from each other, as well as the way we're all the same. And with that, I think I'll quit talking. I put that website up there. I'll put all these slides on my website as soon as I get to the internet, and so you can just download the whole package, and I'll put more references on here to some of these papers I mentioned. So thank you very much. I'll quit talking.
Do you have any questions? Should we have some light in the room? How do you get the side curtains? Thanks. Yeah.
There are 2 American populations that are conspicuously absent from your sample. One of them are the highland Andes, which are pretty conspicuous that populations, and the other Afro-Caribbean and Afro-Americans that have a reputation of being cat populations. Do they fit in your genetic picture?
They haven't been, we don't have samples
Have data.
yet. We don't have samples. There may be samples from both of them at Yale, and I may be able to talk Ken Kidd into typing them, but I haven't seen the data. Now, you would expect the Andean pattern to be different. Yes. You expect less of it there, I expect. Yeah.
Any other questions? No?
Okay, I mentioned this morning. I mentioned the work of Jay Belsky.
I've and others have suggested that there's a learning mechanism having to do with father absence that determines whether a male is a good parent or a good competitor, and there are effects on females too. I think that learning mechanism is there, and the reason I think it is that if you're raised in a father-absent household because your mother was widowed, the effects are mirror imaged. So I think the learning is there. On the other hand, if we have a 1,000 years, put anyone for 1,000 years in Highland New Guinea, where the only way you get your DNA into the next generation is being a tough guy and a good spear thrower and a good fighter, if there's any genetic diversity, it's going to be selected. So I suspect the ecology changed the 7R frequency. Now, whether that 7R frequency is going to make it hard to change the ecology back, I don't know. It's possible. But I don't have a belief. So I don't have an answer to your question.
Bill?
If we read ethnographies, we find that behavior can change over a year. The police come in and they say, all right, no more fighting. Ideology can then change over 100 years. I think things can get out of sync. I've never been a big fan of trying to do cross-cultural statistics because I never knew how to do it. I think what this stuff needs is someone actually looking at children, and we're writing a proposal now with some people at Irvine who have a sample of several hundred children that have been genotyped in an ongoing study. The problem so far is that the children have been put in two groups. Either they satisfy strict criteria for ADHD, and you got to be a really rotten kid to satisfy that, or not. And the proper thing to do is look for differences between children bearing or not bearing the genes. And I suspect that will be much richer information than this. I presented material suggesting that they aren't run-of-the-mill ADHD kids. They don't have the neurological deficits. They don't have the IQ loss. So I'm not answering your question because I don't have an answer for that. Bad.